Welcome to the lecture series Cell Communication. I'm Janine Post of the Developmental Bioengineering Group of the University of Twente. In this lecture, I will discuss enzyme-coupled receptors. This lecture is the last of 3 lectures on the most common receptor types, enzyme-coupled receptors. The other lectures can be found on my YouTube channel. Illustrations in this video are based on the book Essential Cell Biology, unless otherwise noted. The information given in this lecture can be found in Chapter 16. To understand this lecture, you need to know the principles of signal transduction as discussed in the first lecture. The bare basics are that an extracellular molecule or ligand binds to a specific receptor, thereby activating intracellular molecules that influence many cellular processes, including cell differentiation, migration, proliferation, etc. Enzyme-coupled receptors are a group of transmembrane receptors that have either intrinsic tyrosine kinase activity in their cytoplasmic domain, or they interact with another protein that acts as an enzyme. The group of receptors with intrinsic tyrosine kinase activity are called receptor tyrosine kinases, and this group is much smaller than the GPCR family, with around 60 unique receptors. Enzyme-coupled receptors were discovered because of their role in cell growth, proliferation, differentiation, and cell survival. Receptor tyrosine kinases bind polypeptide ligands, such as growth factors, cytokines, and hormones. These ligands and receptors act locally at very low concentrations, of the pico to nanomolar. Responses are typically slow, in the order of hours, and require many intracellular transduction steps that eventually lead to changes in gene expression. Abnormalities in signaling via receptor tyrosine kinase and other enzyme-coupled receptor play a major role in the development of cancer. The basic steps of the RTKS signaling pathway are highlighted here. First, you have ligand binding, such as growth factors, which induces receptor dimerization and phosphorylation of its cytoplasmic tail. On these phosphorylated sites, adapter proteins can bind, and they can activate intracellular signals, for example the RAS protein. In turn, RAS activates kinase cascades, and the last kinase activates a transcription factor. In addition to the slow response resulting in changes of gene expression, enzyme-coupled receptors can also direct rapid responses, changes in cytoskeleton, changes in cell shape, and the way the cell moves. These changes occur in seconds after addition of a growth factor, as is shown in this movie, where a red growth factor is added to green colored receptors. A kinase is an enzyme that transfers a phosphate group of ATP to a free hydroxyl group in the side chain of a serine, threonine, or tyrosine. In this example, the phosphate is added to a serine side chain. A phosphatase catalyzes the removal of this phosphate group. Interestingly, much more work is being performed on describing kinase function than on phosphatase function, even though both play an important role in tuning the signal strength and length. Both of these aspects are often seen to be misregulated, leading to diseases such as cancer. The terminal phosphate group of an ATP molecule can be transferred to a hydroxyl group on the side chain of an amino acid, hereby forming a phosphoester bond. You can read more on the chemical reaction and properties of ATP in Chapter 3 of Essential Cell Biology. Amino acids link together by peptide bonds in which a hydroxyl group is lost from the carboxyl group of one amino acid, along with a hydrogen from a second amino acid, thereby forming an amide, which we, in this case, call a peptide bond. In polypeptides, we therefore distinguish the backbone, here in gray, and the amino acid side chain, here in the color. The hydroxyl group of the C-terminus are needed for making the backbone and therefore not available for phosphorylation. Instead, kinases transfer a phosphate group to those amino acids that have a free hydroxyl group in the side chain, as shown here for tyrosine. So in contrast to other amino acids, such as alanine, threonine, serine, and tyrosine contain a free hydroxyl group in their side chain. You can read more on the properties of amino acids and polypeptide chains in Chapter 4 of Essential Cell Biology. In order to tune activity of signaling pathways, proteins can be phosphorylated. And this phosphorylation can either increase, as in this case, or decrease the activity of the protein. This depends on the site of phosphorylation and the structure of the protein. Signaling molecules that bind to receptor tyrosine kinases are often dimers, shown here. Binding of this dimeric ligand causes dimerization of two receptor molecules. This causes a conformational change, which brings the cytosolic kinase tails closer together. This allows the kinases to phosphorylate the adjacent tail on several tyrosines. Each phosphorylated tyrosine functions as a docking place for an intracellular signaling protein, thereby relaying the signal. These docking proteins contain an SH2 domain, with which they bind specific phosphorylated tyrosines. They are therefore called the SH2 domain proteins. Examples of this are the oncoprotein SARC, which was the protein the SH2 domain is named after. SARC, homology domain 2. After receptor phosphorylation, an adapter protein docks on a specific phosphotyrosine on the receptor tail. This adapter protein recruits a RAS guanine, a nucleotide exchange factor, or RAS-GAF. In turn, RAS-GAF stimulates the inner membrane-bound RAS protein to exchange its GDP for GTP. In contrast to the trimeric G proteins that we've discussed before, RAS is a member of a large family of small GTP-binding proteins called monomeric GTPases. After a delay, RAS is switched off by RAS-GAP, GTPase activating protein, which promotes hydrolysis of the RAS-bound GTP to GDP. Most receptor tyrosine kinases activate RAS, including fibroblast growth factor, platelet-derived growth factor, nerve growth factor, etc. And as long as the receptors are dimerized, the downstream signal transduction pathways remain active. The signal can be inactivated by either tyrosine phosphatases or by endocytosis. When ligand-bound receptors are internalized via endocytosis, mostly via clathrin-coated pits, the receptors are either degraded or separated from their ligand and recycled back to the plasma membrane as inactive receptor tyrosine kinase, ready to bind new ligands. The RAS protein activates a 3-kinase signaling module of MAP kinases, or mitogen-activated protein kinase, that relays the signal to activate proteins and specifically transcription factors. The first in this cascade is the MAP kinase, or MAP3K. Examples of this are MAC kinase, TAC, and BRAF. MAP3Ks activate MAP kinase. For example, MAC, or MAP kinase ERK kinase. This activates MAP kinases such as P38, JNK, and ERK. MAP kinase pathways are activated by many cellular processes. Examples are cell proliferation, cell growth, and differentiation, but also in DNA repair after stress responses as a result of oxidative stress, UV stress, and inflammation. Extracellular survival signals, such as IGF, activate an RTK that recruits and activates PI3 kinase, or phosphoinositide 3 kinase. PI3 kinase then phosphorylates an inositol phospholipid, which is embedded in the cytosolic side of the plasma membrane. This phosphorylated inositol phospholipid attracts intracellular signaling proteins. One of these signaling proteins, AKT, or protein kinase B, is a protein kinase that is activated at the membrane by phosphorylation mediated by two other protein kinases, which are here called protein kinase 1 and protein kinase 2. Protein kinase 1 is also recruited by the phosphorylated lipid docking site. Once activated, AKT is released from the plasma membrane and phosphorylates various downstream proteins on specific serines and threonines. One of the proteins activated by phospho-AKT is BAD. If not phosphorylated, BAD promotes apoptosis, or programmed cell death, by binding and inhibiting BCL2, which otherwise suppresses apoptosis. When BAD is phosphorylated by AKT, it releases BCL2. BCL2 then blocks apoptosis and thereby promotes cell survival. This example shows that the presence of survival factors is important to ensure that imminent cell death is prevented. In this figure, we see 5 pathways that are activated by GPCRs and or RTKs. Two pathways lead from GPCRs through adenylyl cyclase and through phospholipase C. And 3 pathways lead from RTKs through phospholipase C, RAS, and PI3 kinase. Each pathway differs from the other, yet they use some common components to transmit their signal. All 5 eventually activate protein kinases. The overall effect of the cell's response is therefore dependent on many factors, including the combination of extracellular signals, the types of receptors on the cell membrane, strength and duration of the signal, presence of intracellular signaling molecules, and even the epigenetic memory of the cell. One could see how many diseases are an effect of misregulation of these processes. And this makes studies of signal transduction interesting, but also challenging. Thank you for watching this video. If you have any questions, please respond below or email me. In the next lecture, we will discuss methods for investigating signal transduction activity.